Take 1 milliliter of blood from a completely healthy person. About a fifth of a teaspoon. Floating in it, outside of any cell, are somewhere between 200,000 and 3.7 million whole mitochondria. Not pieces of them. Not loose mitochondrial DNA. Intact organelles, with membranes, drifting free in the plasma. And here's the part that gets me. Nobody knew this until 2020. The researchers who found them said as much. Given how many there are, the obvious question is why this hadn't been noticed before. Today I want to walk you through that finding, the discovery that made it make sense, and then the serious scientific fight about what it actually means. Because that last part is the most useful thing in this video. Let's start with why loose mitochondria in blood were surprising at all. The textbook position, for decades, was that mitochondria are strictly yours. You get them from your mother's egg. They replicate inside your cells. Cells build more of them when they need more. And that's the entire story. They arrive by inheritance or local manufacture, and they stay put. That view started falling apart in 2006. Researchers took a line of lung cancer cells and stripped them of their mitochondrial DNA. Without it, those cells can't run oxidative phosphorylation properly. They're crippled. They can't respire normally and they can't proliferate the way they should. Then they grew those crippled cells alongside bone marrow stem cells. The crippled cells recovered. They regained respiration and they started dividing again. The stem cells had given them mitochondria. Whole working organelles, transferred from one cell to another cell. Horizontally, not by inheritance. That result opened up an entire field. Since then, intercellular mitochondrial transfer has been documented in a long list of tissues, and researchers now describe it as a fundamental mechanism for maintaining tissue integrity when cells are under metabolic stress. So how does an organelle physically get from one cell to another? The main route is something called a tunneling nanotube. These are thin bridges built out of actin, the same protein your cells use as internal scaffolding. One cell extends a tube, it connects to a neighbor, and cargo moves across. Not just mitochondria, but that's the cargo we care about here. In the nervous system, these have been seen carrying mitochondria in both directions between neurons, glial cells and blood vessel cells, and researchers think it's part of how tissue recovers bioenergetically after injury. There's a lovely detail in the mechanics of this that connects to something I covered earlier this week. Before a mitochondrion gets pushed into one of these tubes, it frequently undergoes fission first. It splits into a smaller piece. If you watch the video on mitochondrial dynamics, that's the same machinery, the same protein doing the cutting. There I described fission as quality control, separating damaged sections so they can be cleared. Here it's being used as a packing step. Make the cargo small enough to fit through the tube. Same tool, completely different job. That's very typical of how cells work. Alright. So we have transfer between cells, we have a mechanism, and we have millions of mitochondria floating in blood. The tempting conclusion writes itself. Your body has a courier network. Mitochondria are being shipped around the bloodstream to cells that need them. And this is exactly where I have to slow down, because that's not established, and the argument about it is genuinely instructive. Here's what happened. The team that found the circulating mitochondria in 2020 didn't just count them. They tested whether they were still working. They measured oxygen consumption, and they reported that these free-floating mitochondria were respiratory competent. Still capable of running the electron transport chain. That's a big claim, because a working mitochondrion in your bloodstream could plausibly be delivered somewhere and put to use. Then another group of researchers went back and checked. They used high-resolution respirometry, which is a more sensitive method, and they included a proper control, using mitochondria extracted from platelets in the same blood samples. They confirmed the mitochondria were there. The markers were present. But when they measured respiration, they found the rate was not significantly different from zero. Their conclusion was direct. Circulating cell-free mitochondria are unlikely to be functional in the living body, because they show no capacity for oxidative phosphorylation. So the headline finding got substantially walked back. And if this channel had covered the first paper when it came out, I would have had to come back and correct myself. But here's the part I actually want you to take away, because it's a model for how to think about all of this. That same skeptical team, the ones who found the respiration was essentially zero, did not conclude the phenomenon was meaningless. They wrote that despite probably not being functional in the narrow energy producing sense, circulating cell-free mitochondria may still have significant physiological roles that remain to be worked out. That's the correct posture. They killed the specific claim and left the door open on the general question. And there are reasonable ideas about what those roles might be. Mitochondrial DNA resembles bacterial DNA, which means your immune system reacts to it. So mitochondria released into the bloodstream could be functioning as signals, telling the rest of the body that something somewhere is under stress. A messenger doesn't need to be producing ATP to carry a message. So the accurate summary is this. There are millions of them in your blood, that's not in dispute. Whether they're working power plants is, and the current best evidence says probably not. Whether they matter is a completely separate and still open question. Now, what do you do with this? Nothing. And I want to be direct about that, because this is the point where a different channel would make something up. There is no way to boost mitochondrial transfer. There's no supplement, no protocol, no breathing technique that's been shown to increase it. Most of the research is in cell culture, and a great deal of it is in cancer models, where transfer sometimes helps tumors rather than you. If somebody starts selling you mitochondrial transfer as a wellness intervention, they are far ahead of the evidence. What I'd say instead is that this whole area is fundamentally about repair capacity. Your body maintaining and restoring the machinery it has. And when it comes to actually building that capacity, with real human evidence behind it, the answer hasn't changed and it isn't exciting. Muscle contraction. Movement. That's the intervention with the strongest support for improving mitochondrial function in people, and it's free. I'm not claiming the studies I've described today prove that. They don't. I'm telling you where the solid ground is, next to the interesting ground. As for what I promote, I'll keep it short today, because this video isn't really about supplements. CoQ10 is one of the carriers in the electron transport chain, the structure that makes ATP inside functioning mitochondria. Levels tend to decline as we age. That's the claim. Nothing about transfer, nothing about circulating mitochondria, because nobody has studied that and I'm not going to imply otherwise. If you want to support that machinery, the link in the description below is an affiliate link to Mytelin, which is PQQ and CoQ10, with a 90-day money-back guarantee. And if you want more videos where I show you the study and then show you the study that argued with it, subscribe. That's what we do here. This video is educational only and it's not medical advice. Talk to your doctor before starting anything new.